First of all, I would like to thank all of you, all participants. I'm happy to be here to present my work. Okay, it's related with Isoform and pathway level analysis of melanoma transcriptome. I would like to say that the project is ongoing and there is no clear over the results and interpretations, but I will show you some interesting facts and challenges which we were faced during our works. So basically, a study is about melanoma transcriptome, and let's briefly discuss what melanoma is. It's type of cancer which occurs in some type of melanoma cells, and it's mainly occurs in skin and rarely in different places. So there is 5 different types to classify the melanoma. It starts from simple nevus, simple nevi cells, and if, for example, the border of the nevi is asymmetrical, or the colors are different, size is big, and etc., we can classify that it become from benign cells to the malignant melanoma, that means cancer. And so there is basically 2 type of classification: either it's non-cancer, nevi, or it's melanoma. But it showed that this method of binary classification is not enough for sufficient treatment, and so more cancer subtypes needed, new classifications needed to understand more precisely the stage of melanoma to have efficient treatment. Our colleagues had data of 80 samples, and they tried to extract some subclasses, have new classifications methods which can help to treat this cancer much better than now. So, as we already heard a lot, for that purpose they used self-organizing map clustering methods, so they found basically 2 subtypes for both benign nevis and melanomas. It's type 1 and type 2. So we can see here the transcriptome maps, which are typical for each subtype. For example, melanocytic nevus type 1s have activated measures activated gene in translation processes, ribosomal activity in DNA, while the melanocytic nevus type 1 have endothelial and stroma-specific genes. And then also melanocytic, the melanoma subtypes. Melanoma also had 2 subtypes there, from which one activates pigmentation and proliferation and DNA damage, and it's kind of first stage and good prognosis. And the second type is melanoma type 2, which activates immune response and proliferation, and it's kind of so-called bad prognosis, late stage of melanoma. These all analysis and classifications were performed on gene level, but let's briefly discuss some numbers here. So when we are looking at transcriptome at gene level, we have like approximately from 90,000 to 21,000 genes. Like roughly say we have 20,000 genes. But here is the range because different sources reports different number of genes, and there is no exact number of genes there. But as we know, biology is much complicated than that, and so we have much more complexity in the second level. When genes are transcribed to the isoforms, we have 10 times more isoforms, and coding complexity increases to tenfold, There is now well-reported 200,000 isoforms. Even there is other sources which claims 300,000 isoforms. How many functional proteins they encode is still super debatable, and it was one of the big challenges in our work because there is 200,000 isoforms, but no one knows exactly how much of them have functional phenotypical impact, and are they make any sense or not. For understanding all this stuff, let's shortly discuss also alternative splicing, which locates in between these genes and isoforms, which made this big complexity of transcripts here. Here we have the gene, which contain coding and non-coding parts. Non-coding parts are introns and coding parts are exons. After transcription by RNA polymerase, these RNA undergo alternative splicing, and during this process, these introns are cut out and exons joined together. But this process can go in different ways. I mean, if these spliceosomes put together different combination of exons, from the other side you can get different sequences and obviously different proteins, from which some of them can be functional distinct, or non-functional, have no any function there. So to understand what's going on in transcriptome level, we performed analysis on isoform level, and we went much deeper to understand is there any new information on transcriptome level, on isoform level. And so because the purpose of this work is that in recent times many papers published which related with alternative splicing, and some of them are concentrated in specific genes, and this publication showed that some genes have a really important role in cancer development, which related with alternative splicing with other isoforms. So there is other comprehensive approaches which evaluates the entire transcriptome level, the distribution in human cancers, so many type of cancers. So we have decided to do analysis on these melanoma samples, on these 80 samples, and so this is like our workflow. First of all, we did transcript level alignment with Callisto liner, which allows to try to assess abundance, quantify abundance of transcripts from short reads, and then we use this received data, this isoform abundances, to understand is there any connection between these transcript level expressions and gene level expressions, and gene groups and patterns which our colleagues already calculated. And then we try to understand is there any isoform switches. I will cover this, what is isoform switch after a few slides. And then we try to evaluate the isoforms on pathway level to understand is there any useful and interesting things on systematic level, on pathway level. So first of all, let's shortly describe what is Callisto, what kind of aligner it is. So it's quite different from traditional aligners, and it works much faster and memory efficient because of some specific characteristics of this aligner. It also compatible to perform quantification on isoform level. So briefly, what it does, it does not do exact alignment, but instead it's cut out. It separates the short reads to chimers to small pieces and try to understand the compatibility of these chimers with one or another particular transcript, and it does not perform precise alignment, but it does this all quantification on a statistical evaluation level. For example, we can see here that we have reach, and there is different type of transcripts, and it starts to assign the part of this read, these chimers, to the first 3 points, the parts, and for first section, these chimers are compatible with 3 isoforms. Then after it goes further, it can precisely classify from which isoform it comes from, this particular read. So after performing Callisto level alignment, we received 200,000 isoforms for all samples, and then we tried to understand what's the distribution, what's the connection of these isoforms with already calculated genes. So here is the global overall landscape of melanoma transcripts, which are already calculated by a Leipzig group. And so we can see here that it's colorful map and have some red spots, which contain some microclusters of genes, which are related with some processes there, and with these spots we can do some classifications of melanoma subtypes, and so we can see that, for example, G spot related with DNA damage, spot A have gene sets which connected to translation, B with pigmentation, etc. So we try to correlate our isoform expressions with these metagenes, with these microclusters, to understand what's the distribution of our isoform expression on the already known gene distribution map. And so we found that, and we looked at the correlation of isoforms from specific spots to the others. And we found that some spots have really specific isoforms, and they does not have any correlation with other processes. So it's not visible here, but we can see that this spot is related with immune response, and it have a really specific set of isoforms. And there is almost no correlation with the other spots, with the patterns of gene expressions from other spots. And so there is really little number of correlation with others. So, but on the other hand, there were spots which had many correlated isoforms with other spots. For example, the G spot, which have a lot of genes involved in these DNA damage processes, they had mutual correlated isoform patterns with other spots. And interestingly, it's interesting that G spot also contains a set of genes by which we can separate type 1 and type 2 subclasses. And these highly correlated spots also had these gene sets by which we can classify these subclasses of melanoma. So after looking at correlation between some particular spot isoforms with the other spots, we try to understand if there is any correlation of isoforms from non-detected part, these non-significantly expressed gene parts, to the significant expression spots. And we found that almost 20% of isoforms, had a correlation with these gene expression spots. And what does it mean? That this part on gene level, there is no any significant changes, but there is isoform alterations there, which pattern associated with these functional sets of genes. And here's some percents. These non-spot areas have correlation with these spots, with the gene sets of had similar gene sets with A spot, B spot, and we can see that some spots, with some spots, they have really high correlation. So that means that we can somehow predict that these non-significantly changed genes contain isoforms which involved in some processes in these spots. So then I would like to show another picture which related with isoform fraction variance. What is isoform fraction? It's just simply the part of expression from gene. That means isoform expression divided to the gene expression. And for example, if we have one gene which contain, I don't know, 3 isoforms, and their fractions are, I don't know, 0.5, 0.2, and 0.3. And we can clearly see here, it's like we took the variance of these isoform fractions and averaged for each metagene on this map. And it is visible that in detected spot areas, these genes almost have no any variance. And that from this can be considered that these genes contain only single isoform and there is no big variance. But this in non-spot areas, which comes from these parts and this part, have high variance. That means there is some significant alterations which might have some functional consequences which needs to be further investigated. So after looking at correlations, we try to detect isoform switches. How we perform that? First of all, let's understand what is isoform switch. Let's say we have one gene which have 2 isoforms, and for example, in a healthy sample, one isoform is highly expressed, but the other is down. But for example, in tumor sample, the first one is down, but second one is highly expressed. In this case, with classical gene expression, differential gene expression analysis, we can't find any significant change on gene level, but still there can be isoform switch there, which can have some consequences from which one can be I don't know, of different functional isoform or non-functional isoform. That means that gene loses its function, but we can't see it in gene level. And so we perform differential expression analysis on gene level, then on isoform level, and then we took all isoforms which had significant change on only isoform level, but not in gene level. That means that this isoform have alteration on isoform level. One isoform goes down, went down, and the other up, and there is no change on gene level. And what we saw that there is in subtype differential analysis, we found 182 switched isoform in melanocytic nevus versus melanoma, and almost 800 between type 1 and type 2 subtypes. So, and 3 of them, switched isoforms was related with was come from set, and this it was shown that this set of genes had functional consequences in different types of cancers. So we can see 2 isoforms from type 1 versus type 2 analysis and 1 isofrom from native versus melanoma analysis. The most obvious example can be CD44, this switched isoform, which is a surface protein, and we can see that the second isoform had isoform switch significant, and this isoform version is highly involved, highly expressed in melanomas, and it's some surface protein related with cell migration and progression. So it's one of the indicators that some of isoforms can be really important switch isoform for analysis. So then after detection of switched isoforms, we try to understand is there any relation between switched isoforms and pathways. So we took the model of pathways from K. These pathways are logically separated in different processes, and so they start from receptors and ends with some molecular processes. And so we try to understand the isoform switch distribution on signaling pathways. And so we see that some signaling pathways had really many isoform variants inside their nodes, and we can see that some of pathways are related with melanoma, and they are in the top places of isoform complexity there. After that, we performed some analysis to understand are there many isoform switches on the pathway nodes, which are very important. We measured the importance of node by degree centrality, which is like the number of interactions of that particular node, and we can clearly see that the nodes which have high centrality have more isoform switches than the nodes which have low centrality. That means that high centrality nodes can have more isoforms, and they have potential to have isoform switch there. It's not visible here, and we looked at isoform quantity for nodes which have inhibitor interactions and switches, and we can see here clearly that the nodes which have inhibitor interactions and switched isoforms, their isoform quantity is much higher not much higher, but there is some difference between non-inhibitor and nodes and the nodes which don't have isoform switches. Pretty much that's it about our analysis. We ended up with this stage, and there is some further works to be performed. One is to understand the pathway activity state based on these isoform expressions. That means to simulate some pathway activity analysis with different combinations of isoform expressions to understand is there any space of activities depends on patterns of isoforms. And then in the further step we will try to functionally annotate these switched isoforms to understand more precisely what's going on in functional level. So, and many more. So with this, I would like to thank our group and ISB group. There should be their picture, but I didn't manage to take picture. So thank all of you. Thank you very much. So.